Welcome to the inaugural lecture of 2024-2025 DeepMed Seminar. The DeepMed Seminar is a monthly history forum for science, strategy, and the environment. You can find the complete calendar in the DeepMed website. This year we have two main novelties. The first one is that we will focus on the Mediterranean Sea, with talks about this maritime space by experts from very different disciplines, from history to oceanography. Second novelty is that all of our speakers will visit us in Sevilla. So we will keep the hybrid format so that we can enjoy the conversation with colleagues far away, but we will also have the luxury of talking to speakers both before and after their talks. And on this occasion, I have the honor to introduce Isabel Cacho, who will be offering this inaugural lecture on decoding past changes in the Mediterranean deep sea. Isabel Cacho is a geologist, but belongs to two relatively rare subspecies. One of them is paleoceanography, and the other one is the users of geochemical tracers. And today she will explain in detail the connection between those two things. She works at the University of Barcelona, and she has published on climate variability in the Mediterranean and elsewhere in journals including Nature, Science, and all the usual suspects. So Isabel, thank you very much for coming, and we are really looking forward to listening to you. Thanks.
Thanks very much. First of all, I want to thank this invitation. This occasion for me is really, I really appreciate the opportunity to be here in Sevilla, but particularly to meet this, for me, very exotic group, because I'm more familiar to talk with paleoceanographers, more on proxy data, geochemical tools, and today I was listening to about philosophy, history, social sciences. It's very, very exotic for me, but I'm learning a lot. So thanks very much for all your inputs, and I hope that we can keep going on. And now I'm going to start the presentation. Can I see that? Okay, so I hope you can see so the title is Decoding Past Changes in Mediterranean Deep Circulation. So in some way, I understand that all of us, we share the interest for the ocean dynamics from different perspectives and time scales. But we are a bit interested not only in the surface of the ocean, that most of the times we keep just looking at the ocean in the very surface part, but also we are interested in this deeper part. And so what I'm going to explain to you is the strategy that I study the ocean. I have to say that I did my degree in Barcelona as a geologist. And when I finished my degree, I moved to Germany, to Kiel in the north. And there I started to work in a laboratory of organic chemistry. And that everything was very, very exotic for me. But the application of that organic chemistry was to learn about the ocean in the past. So paleoceanography, it was called. That was absolutely new for me. It was the very first time I heard about that. And I was fascinated. And I decided that I wanted to keep going on this discipline. So I started to do a sort of master. At that moment, it was called Tesina, within Germany and Barcelona. And then I could do my PhD on paleoceanography. And from that, I had the occasion to go to Cambridge University with one of the fathers of the paleoceanography, which is Nick Shackleton. And so I moved from the organic tracers to the inorganic ones there. I learned new techniques, and I have been always trying to incorporate new techniques, and that's what I'm going to talk today. I'm going to show you different proxies, different tools that we apply to trying to reconstruct the interior of the ocean. So I'm sorry. No. How do I pass? Okay. So probably you don't really know this introduction slide. I think you really know very much about that. Actually, I have realized that you have this. This is actually this picture here on the wall, so you know it very, very, very well. I don't really, but I would like to highlight that this Mediterranean circulation is really controlled by the climate, and this region is really like the boundary region with two different climate systems. One of the intermediate latitudes and the low latitude system. We have a big continent in the north with a relatively humid climate, and a big continent in the south with a really arid climate. And these climate conditions are those controlling the properties of the Mediterranean waters. Actually, we know very well that evaporation exceeds precipitation and runoff, and that controls the formation of these water masses that they are very well represented in this diagram. So here we have the inflowing water mass coming from the Atlantic Ocean, and these climate conditions control the formation of this intermediate water and this eastern and western Mediterranean the water masses that permanently end into the Atlantic with this small Mediterranean outflow water. And I will talk a bit about all these water masses. But what is very important also for me, and I will mention very shortly in this talk, but I think that we have to keep in mind that these water masses, these deep water masses, intermediate water masses, are formed in very specific basins of the Mediterranean. We have the intermediate water mass formed in the Levantine Sea, and the deep ones are formed in the Adriatic and the Gulf of Lyons, in the case of the Eastern and the Western Mediterranean. But we know now that the Aegean also has an important role forming these water masses and particular periods. So one of the motivations for me in my research is also trying to understand the individual role of these convection cells. In the past and in the future, often is told that the convection thermal circulation in the future is going to become weaker. And we know that in the past has been sometimes stronger, sometimes weaker. But we don't really know the behavior of individual cells. And what is actually, if they are responding in the same way or in a different way, if there is a migration from one to another. So that was a bit of my motivations in some of my research. I will mention this very, very briefly. So, yeah. So as you know, Mediterranean Sea is very sensitive sea, it's very small, so changing the properties is easier in the Mediterranean than in the open ocean. And there is several studies that they are actually showing this here. Sorry, now. Here we have the classical ST diagrams where we have the salinity and the temperature. We define the properties of water mass. Densities are illustrated here with these lines. Oh, sorry. Okay. And so we can see in colors changes in the properties across time. And that's in a particular region in the northwestern Mediterranean Sea. And what we can see in this region is that the conditions along time, these water masses here are the deep water masses, are becoming warmer. And in surface, these are the surface water masses, are becoming even larger the changes. They're becoming saltier and warmer. So it's reacting. I mean, we see in different locations how the Mediterranean Sea is already reacting in surface, but also in the deep water mass to this climate change. And when we go to projections, future projections, there are different models that tell us that by the end of the century, is expecting to have a weaker convection. Here in this diagram, we have with colors the bedrooms, and they will indicate a stronger overturning. And here we have weaker, that will be the situation for the end of the century. So we know that's very sensitive. We are expecting these changes, this circulation to change in the future. But actually, we know very little about what is the natural range of variability of the system, how it behaves to natural climate changes, and whether these convection cells, individual convection cells, have the same sensitivity and react to the same forcings. And we have expect that all of them, they have a simultaneous response or not. So these are some of the open questions. And I tell you, I'm not going to answer to every one of these. We do small advances, and we know a bit more, but still many of these questions are open. Now, what my main source of information are deep sediments, marine sediments. This is let's say the source of my data. My library, the way I study the ocean is with marine sediments. And here you have in this image a sediment core, and actually the sediment core comes from the Eastern Mediterranean Sea, and you will see that they have a lot of color changes. And I have chosen this Eastern Mediterranean core because they are lovely sediment cores in the Eastern Mediterranean. They have a lot of color changes. But when you go to the Western, they are really ugly, gray, boring. You don't really see many changes. But when you analyze them, you see the changes. But visually, you don't see. And what we have here in black is what is called the last sapropel. I'm not sure whether you are familiar with this term, but if you go to the paleoceanographical literature, the last sapropel is very, very much studied a lot. And actually, I was mentioning before, when I started my PhD, I couldn't find a single publication on the paleoceanography of the Mediterranean, which was not dedicated, or an important part of it was talking about the sapropel. Why? Because the sapropel was a fascinating moment in the circulation of the Mediterranean, and that made that everyone in the paleoceanography was very much focused in the last sapropel. But many other things have happened in the Mediterranean, as you will say. But first of all, I want to talk you a bit more on this sapropel. It's black. It's black because it is a layer rich in organic matter. It's a moment, it's a large accumulation of organic matter. And why? It's because this deep convection that we saw before was stopped. We didn't have this deep convection. So that led that the deep water in the Eastern Mediterranean Sea, in the Eastern Mediterranean, became almost disorcic, means no oxygen, so preservation of organic matter. And the sapropel, it was the result of a major change in the properties of the surface waters. Waters become fresher because the pluviosity, here we have 2 diagrams for the African continent, and we see on the left a situation comparable to the present day when we have humid conditions towards the equator, more towards the equator, and very arid conditions in the Sahara. And you look to the right, we see a situation very different, where these humid conditions extend northwards. So at this moment, there was the early part of the Holocene, which is the interglacial where we are nowadays. The ITC set, which is this convergence zone where maximum precipitation, where the monsoons concentrate in the African continent, was displaced northward. And that made higher precipitation to reach higher latitudes, and rivers like the Nile, but also rivers that nowadays don't exist, they were flooding fresh water towards the Mediterranean Sea. So that changed the salinity of the Mediterranean Sea and prevented deep convection, and led this last sapropel form in the early Holocene. So this was a fascinating moment in the circulation of the Mediterranean. Many of the researchers is focused, even though as you say now, we see there's other changes very interesting. Now here, it's a bit complex figure, but I look at you a bit on the time period I'm going to talk. Here we have in red the temperature record. This was from my PhD, a sea surface temperature record done with organic biomarkers, and we see changes in the Alboran Sea. And we see this is the last glacial period, the last deglaciation. Here we have a back to cold conditions. That's the younger dryas. And I want to mention, I'm going to talk a lot about the younger dryas. So remember that was in the middle of the deglaciation. It was a period where conditions went back to cold. And then we have the Holocene period. Here, if we go to the lower part, we have a proxy which shows the sapropel, which actually have two phases, the sapropel 1A and 1B. Okay, and that happens in the Eastern Mediterranean Sea, as I mentioned before, due to this change in the ITCZ driven by orbital changes. But in the Western Mediterranean Sea, also occurred a period of accumulation of organic matter, which is not so well known, is what we call the organic rich layer 1, that occurred in the Alboran Sea. And we observed that it started far earlier than the sapropel period that occurred during the termination. And we have proposed in some papers that is linked to sea level changes, the freshening of the surface layer due to the sea level rise, that led to the waters of the Western Mediterranean to be more stratified and form this organic rich layer. Was not so rich in organic matter as the sapropel. It was not so dysoxic as the sapropel, but there was enhanced preservation of organic matter. So one of my motivations on my career has been to understand whether this Eastern and Western basin, they were operating independently for these events, or there was a connection. And for that, I realized that we need to look for new proxies, new tools to understand better the interior of the ocean. So I'm going to show you some of these tools that we are working. So during my presentation, I'm going to talk about 5 different tools. One dedicated to understand changes in the oxygen content. The other is going to be dedicated to understand, mixing rates within different water masses. I will explain more in detail later. The other is going to be dedicated to understand more residence time of a water mass, means the time that a water mass is in the interior of the ocean. The other is going to be dedicated to understand intensity of the currents. And the last one is going to be dedicated to understand the classical parameters, which are the physical properties of the water mass, salinity, and temperature. So we see some examples, okay? So let's start with the first one, with the oxygen content. A water mass, the oxygen content of water mass is going to depend on 2 variables, the time, which has been insulated of the atmosphere. A water mass is incorporating oxygen by exchange with the atmosphere, and exchange with the atmosphere, and thanks to the photosynthesis is the way to incorporate. At the moment that a water mass enters into the ocean, there is no more photosynthesis, there is no exchange. So oxygen should be constant, but since occur many processes of organic matter oxidation, this oxygen is getting lost because there is a lot of oxidation processes in the water column. So in some way, we can do oceanography with the oxygen. And here you have a transect. This is a transect. Here we have this is a cruise from a project. We have studying the outflow of waters coming from the Mediterranean, here indicating the path through the Strait of Gibraltar, and there's the path through the Iberian Margin. So this part here is the Mediterranean Sea, and this part here is the Atlantic. This will be the Gibraltar Strait, okay? The Gibraltar Strait. And the colors indicate oxygen content. And the red colors mean more oxygen. The blue colors indicate less oxygen. So you can see that we can, in some way, do oceanography. We have a minimum oxygen related to these Levantine intermediate waters coming from the Eastern Mediterranean Sea. Western Mediterranean deep waters are more oxygenated because they generate in the Western Mediterranean Sea. So they have been close to the atmosphere in less time. Levantine intermediate water has done a longer path, and then this path coming from the Eastern Mediterranean is lost in oxygen. But look at that, when you go to the Atlantic, really the oxygen minimum is detected through the Atlantic waters are far richer in oxygen. So that was to convince you that looking to the oxygen, we can try to understand a bit the oceanography. So now let's go to also I wanted to show you, if you look to modern oceanography from the Mediterranean, we can see that in this ST diagram, again, with colors, we have oxygen, and we can see the different water masses. Again, they have Levantine intermediate waters, they have the minimum in oxygen. And here we have another study where during since 1994 to 2014 is studying processes looking when events of deep convection has occurred in the northwestern Mediterranean Sea. And we can see, for instance, here a very strong event of deep formation that it changes the properties, the salinity, also the oxygen of the water mass. So really the oxygen content can tell us a lot about the oceanography. So how do we look to the past times with oxygen? So we are using a proxy that is measuring the uranium manganese ratio in the overgrowth crust form on tests, biogenic tests. I mean, the first diagenetic process is when the rest of organisms fall in the deep sea. There is a diagenetic process that very immediately start to form a cross rich in manganese and iron and incorporates uranium. But uranium and manganese have a completely opposite behavior in front of the oxygen conditions. If there is a low oxygen, uranium fixates more. It is a high oxygen, manganese fixates more. So looking to this ratio, we can try to reconstruct changes in oxygen. And this pretends to be a pseudo calibration curve. I mean, this large error, but we can see that uranium manganese increase when we have lower oxygen conditions in the waters. So this is going to be our tool to try to go to the past. And now I'm going to show you an example. This is a bit complex figure, but you see the last 20,000 years. Here we have this deglaciation, and you see a lot of colors. The different colors correspond and sediment cores from different depths. All the pink or violet colors correspond cores from the Alboran Sea, but at different water depths, from more than 2,000 meters to 300 meters. So a wide range of depths. And what I want you to see is that during the deglaciation, during this organic-rich layer, you see very high values of uranium manganese, indicating very low conditions, really very low conditions, particularly in the intermediate waters. So it was a signal coming from the Eastern Mediterranean Sea. But what I want here to focus is you look to the younger dryas, this period in the middle of the deglaciation, look at this major drop at 900 meters. We have a strong ventilation event at 900 meters of the water mass at this depth. Something happens very strong that affects particularly the intermediate water mass of the Eastern Mediterranean, also a bit the deep, and not so much the very shallow 300 meters. So we don't really know what, but we will see more with the other proxies. I also want to show you here, this is the last subpropel in the Eastern Mediterranean core. It's a core from here, from the Adriatic Sea, and we see the same proxy where this subpropel forms. So in the eastern, we have very good ventilation in the deep layer during this organic rich layer, but right when the subpropel was formed, conditions in the western, they were very, very well ventilated. So really the two basins, they were operating in a very opposite way. But I want to learn a bit more about what happened during these younger dryas. Okay, so we are going to go to the second tracer, which is a proxy for the source of the water masses or the rate of exchange of water masses, which is one of the main objectives of my career. Has focused very much in developing this proxy in the last few years. Actually, I mentioned that many of these results come from a ERC that I had, and one of the objectives of this ERC was to set up the labs to be able to apply this technique. And this technique is the measure of neodymium isotopes, first in waters and secondly in the sediments. And here you have the results of a study that we published in 2020, where what we did in a cruise coming from the Eastern Mediterranean Sea, we crossed the Sicily Channel and we went through the Tyrrhenian Sea in this cruise. And one of the things we did in this cruise is sampling waters sampling waters to measure neodymium isotopes and other rare earth elements. Neodymium is a rare earth element. So we work with concentrations of rare earth elements, but also the neodymium isotopes. And why? Look at here, these are the results of this study. This is salinity. And this is a profile, a deep profile. Here we have the depth, 1,000, 2,000, 3,000 meters. This is the state of the state of Sicily, and here we will have the Strait of Corsica here. Okay, and we have the Eastern Mediterranean here and the Western Mediterranean here, and here's salinities, and we can see the entrance of this water mass coming from the Eastern Mediterranean Sea, the Levantine intermediate water with high salinity, and here we see a similar plot with neodymium values. We can trace with neodymium values, but we can also trace with rare earth elements. But with the advantage with these chemical tracers, we can estimate with high precision not high, but at least higher precision, the exchange, the amount of water exported from the Eastern Mediterranean towards the Western Mediterranean Sea. And why? Why we can do that? So the reason is the geology. And here's when the geology has combined with the oceanography to make a really very useful tool. Neodymium isotopes are controlled by the geology of a region. It means that neodymium arrives to the ocean carrying the signal, neodymium signal of the stones, the kind of geological rocks, the nature of the geology in the region, okay? But according with the age of these rocks, the neodymium composition will be different. The very, very old rocks are going to have a neodymium isotope signature which is less radiogenic. When they are very new rocks, volcanic rocks, very new rocks are going to have a very, very high radiogenic signal. When water mass is formed, it means when water mass which is in the surface sinks and enters in the interior, in this convection process brings the signal of the basin, this geological signal with isotopes brings in the interior of the ocean and behaves as a conservative proxy because the residence times of the neodymium is about 1,500 years. So we have a semi-conservative chemical proxy or tracer, and we have the lag that if you look to the geology of the Mediterranean, is very different. Here we have the colors, the neodymium signature. Different colors means different signature of neodymium. So we see that it changes a lot if a water mass is formed. Where is the mouse? It's okay here. In the Aegean Sea, there is a signature different of the Adriatic Sea, and it's different of the Western Mediterranean Sea. So we can trace the source of this water mass according with the neodymium signal. And the good thing for us is that we can do that for current oceanographical conditions, as is the example I'm showing here, but we can also measure the neodymium in the fossil record. So we can apply that in the past time. So this is actually the lab we set up to be able to apply this proxy. This is a Pantalasas, which is the mass spec that we use to measure the neodymium isotopes. And everything, the chemistry needs to be in a very clean condition. So this is the little lab that we set up to be able to do these measurements. We are very proud of these infrastructures. And now I'm going to show you an example in the past. I showed you before an example in the current oceanography, and now I'm going to use the same proxy to apply for the past. And we're applying that proxy in a core located here in the Sicily Channel, very close to the transit we were using before. And here is the record. This is the time record, 14,000 years to present. And these are the neodymium record. And we can observe a period of time with a very high signal, and that has been traduced to percentages of Eastern Mediterranean source waters. It means the percentage of Eastern Mediterranean waters export in this area, in this location. So we can see that during this period of time, younger dryas, we have a very high export rate, of the order of the 57%, compared with the current present day exchange is almost double the exchange that nowadays. On the other side, we have the isopropyl time where we have a very low exchange. So that's the very first example that we can quantify. We can quantify the percentage of water which was exported from the Eastern Mediterranean during the isopropyl time. But the big surprise for us, because we were already expecting to have low percentages during the isopropyl, but what we didn't expect is this very high percentage during this period of younger dryas. So this figure here was to represent the different behavior during the younger dryas, where we have a very strong exchange, a very strong exit of this sort of Levantine intermediate water, Eastern Mediterranean source water, as we call it. And during the isopropyl, this will be very much smooth, very reduced. There was always some exit. I mean, there was always some exit. No, it was not a complete disappearance of this Eastern Mediterranean source waters, but was far weaker this exchange. So if we compare, now I'm going to go to the previous figure that I used before, and I'm going to use that figure during the whole presentation. This is the same records than before. I showed you which was the uranium manganese, and I was mentioning this period of time, younger dryas, where was this reventilation. And look at here. This is the neodymium record I just told you now. This neodymium record comes from here, okay? And we see this period of time where we have enhanced outflow of Eastern Mediterranean source water. So, and we hypothesize that the source of this reventilation, the cause of the Western Mediterranean Sea intermediate waters reventilate, were linked to what happens in the Eastern Mediterranean in some way. Was waters that we hypothesized that coming from the Asian Sea, actually, because this very radiative signature, we believe, comes from the Asian Sea. Now, I'm going to complete this information with another proxy, which tells us about the residence time. What means the residence time? A water mass, when in the surface, is exchanging CO2 with the atmosphere. And atmosphere, the CO2 incorporates what is different isotopic carbons, and there is one 14 carbon, which is an unstable isotope, which is formed due to cosmic rays that act in the in the upper part of the atmosphere. So in the atmosphere, we have this here, 14 carbon isotope, which exchanges with the ocean. Okay, this exchange is not perfect or is not so quick. That means that there is always different signal in the surface ocean than in the atmosphere. We have a bit older signature because it takes a time to compensate or to equilibrate with the atmosphere. But at the moment that a water mass enters inside the ocean, inside the Mediterranean, there is no exchange with the atmosphere. So that means that is no new carbon-14 arriving to this water mass, and this clock is closed. There is no new carbon incorporating, but if this water mass, the longer is inside the ocean, the more this carbon-14 degrades and the older it becomes. So it's a way, if we know what is the carbon-14 of this water mass, we can know the age of this water mass. Nowadays, you can do that. We can actually measure the carbon-14. Well, nowadays we cannot do it because all our nuclear exercise, we have destroyed the carbon-14 signal. So we cannot use now the carbon-14 today, the waters. But we can do in the paleo record, we can measure the carbon-14. If we have some other tool to know what is the actual age, no, I didn't explain well. Okay, imagine we have, for instance, we have this coral reefs forming, these I call corals in the Mediterranean Sea. And when they incorporate carbon to produce the structure, they incorporate the carbon for the deep ocean. So that carbon is going to be old. It depends on the moment. But if we have another way to tell us when this carbon has been formed, another clock, which is independent of the residence times of the water mass, we can measure the difference and learn how was the residence times of this water mass. And with the corals, we can do that using uranium-thorium. Uranium-thorium is another clock, is another geological clock. It's independent of the carbon cycle. So we can date corals with uranium-thorium, the absolute dating, independently of the carbon cycle. And then we can date also with the carbon-14, which is depending of the carbon cycle, and is depending of the reservoir age of the water mass. So with these two absolute dates, we can identify if the residence time of a water mass in the Mediterranean or somewhere else in the ocean has changed in the time. So now I'm going to try to show you the results. And here, I'm using the same graphs that before, where we have the uranium manganese, we saw before, we have the neodymium here, and here we have a series of records showing a drop here, which is a drop in the reservoir ages. It means that we have here values comparable to the present day age of a deep water mass in the Mediterranean, which is around 300 years, is what is the age estimated in the Western Mediterranean Sea. But we here, in this period of time, we have ages which go down to 1,500. Really, that means that there was periods of time that the water masses, they were far longer inside the Mediterranean Sea. And you will say, that happens to be, that minimum reservoir ages happens to be when we have this more exit of Eastern Mediterranean water mass. And we are interpreting that this enhanced water formation in the Eastern Mediterranean is pumping out all water masses that they were in the Mediterranean. They are pumping out through the Strait of Gibraltar towards the Alboran Sea, because these corals are here in the Alboran Sea, about 300 meters are these corals, bones, and they are pumping this old water mass out of the Mediterranean. So we are detecting a very old water mass that is exiting the Mediterranean. But what's important to see as well is a water mass has been in the interior of the ocean for so long time, can be more acidic because more carbon is degraded organic matter. So look, one thing that was not able to explain before is why these coral reefs, right during the younger dryas, these blue bars here indicate growth of these coral reefs. And we see that right in the younger dryas is the minimum growth of these coral reefs. We interpret that this water, very old water masses existing in the Mediterranean, they were very acidic waters that were dissolving this coral reef, preserving the coral reefs. So there was a major change in the chemistry with consequences in the preservation of these ecosystems or the development of these deep ecosystems. Okay, and now I'm going to talk another proxy, another indicator of changes in the deep ocean, which is grand size measurement to identify changes in the currents. Here we have a picture of the Gulf of Lyons. Probably you have recognized. Here we have the Gulf of Lyons. This is the platform. Here we have the Pyrenees. Here we have the Tramontana winds, which are the responsible for the formation of a deep water mass, and that produce a deep formation in the center of the basin here, but also cascading mostly from some of these canyons, and that produced enhanced currents. And we did an experiment it's hard to connect our proxies to the real processes or the actual processes occurring nowadays. But we did an experiment here comparing sediment particles, the size of sediment particles during a year, where we could see events of cascading or formation of the water masses, and the sedimentary record. And what we observed, this is a grand size distribution, that when we have deep water formation, we have quarter particles. When we don't have this deep water formation, we don't have these quarter particles. So we can use a proxy. We have been studying very much this area here, where these deep currents are sensitive, and we can relate the existence of deep currents with the high quartz particles with the formation of the water masses. So this is one of the proxies we're using. But now I'm going to show you an example coming from the Gulf of Cadiz, because links very much with the previous graphs. I have here the same figure than before, with the uranium manganese ratios, the neodymium than the before with orange, these all water masses that we saw before, and adding here green line, which are the particle size, green size measurements here in the Gulf of Cadiz, in an area which is sensitive of this MOW, of this Mediterranean outflow water. In some way, it's telling about the speed of the outflow that's coming out through the Strait of Gibraltar. And I would like to show the good correlates that they're having, this, even though that they are different. You have to see that the orange score coming from here, Sicilia, from this depth, I'm comparing with a core here from the Gulf of Cadiz, but we can see that this increase in the younger dryas is detected with a stronger outflow. This weakening during the subpropel is detected with minimum speeds. So we can really, we're observing that what happens in the Eastern Mediterranean Sea, very likely in the Aegean Sea, has a strong control of the properties of the water masses existing in the Strait of Gibraltar. And the last proxy I wanted to show you is this proxy dedicated to the classical properties, temperature and salinity are the classical properties, but probably the most tricky sometimes to reconstruct. We can reconstruct temperatures using magnesium calcium ratios in foraminifera shells. Foraminifera are zooplankton specimens. They are planktonics, but also they are benthonics. So we can reconstruct temperatures in surface waters, deep thermocline waters, but also in deep waters. It has a relationship, an exponential relationship with temperature. Here, with more temperature, we have more magnesium calcium. This is a thermodynamic response. So this is the tool we are using to reconstruct temperatures. And when we combine that with oxygen isotopes, this is the last proxy I'm talking about, but the oxygen isotopes is the classical tool we use in paleoceanography. This is the basic tool that led the starting of the paleoceanography in the 70s. And oxygen isotopes measured in foraminifera, they are telling us the relationship with the 16 and 18 isotopes, and are telling us changes in the water cycle. When we have the ocean becomes depleted in the light isotope, when we have the building up of of continental ice, of the ice sheets, becomes lighter when we have the melting of these ice sheets, but also oxygen isotopes is sensitive to changes in the temperature of the waters, and also in the regional changes of precipitation and evaporation. So when we combine isotopes with magnesium calcium, we start to have a sort of proxy of salinity. And very briefly, because I think it's already late.
It's all right, you're okay.
Yeah, it's already, but very briefly, I want to show you a very recent example we are using in the Alboran Sea, tracing changes in the salinity using this combination of proxies at different water masses. A depth where we have the Western Mediterranean water mass, a depth where we have the deep thermocline, and at a depth we have this depth sensitive to the Levantine intermediate water. And we have a very different pattern. And I'm not going to go to the details. I want to show you the interpretations here. Here we have a seamount where the formation of these deep sea corals. And when we are saying along the time from the younger dryas to the present, important change in the structure of these water masses, and actually the presence of Western Mediterranean source water very different to what we know nowadays, the Western Mediterranean deep water mass, this water mass during the subpropel actually was bathing mostly the intermediate depth, so a very different oceanography to nowadays. And then after the subpropel, we have the classical structure we have nowadays with these intermediate Levantine intermediate waters and this Western Mediterranean water. But we claim that during the past times and during climatic moments different to the present day, we have a very different structure based in these oxygen patterns and based to these property patterns. And now, as I know that some of you are more interested in time scales more recent, for us it's very hard to work in the last centuries because the resolution of our records are difficult. But I wanted to show you this is a study we published for the last 2,000 years. These are ages in calendar ages. This will be our 2000 year, okay? And here I'm showing two records which are marine records. This is for surface temperatures, and this is for grand size, which is indicator of the speed of currents coming from the Gulf of Lyons. So we have some changes that we interpreted. We have related to the Roman period, to the medieval climate anomaly, to the Petita Lia, the last ice, the little ice age. Thank you. We can differentiate two phases. Here, I would like to stress this record here because this is no marine record. This is coming from stalagmites from Mallorca, where we can actually work in a very high resolution, and we are interpreting this record as an indicator of drier or wetter conditions in Mallorca, in a Mediterranean side, with a strong hiatus here that we interpret as a very dry conditions. And nowadays, we are trying to work at the edge of this record. We are working nowadays trying to connect this proxy record with the instrumental record from the airport of Mallorca, which is going, that's a work in process now. But look here, 1963, 1955, we are dating, this is a speleotherm record. So we are now getting to the stational signal in this site. And now we are in the process to connect this with the actually instrumental record to be able to calibrate our chemical proxy with the instrumental record and trying to do with high resolution the last centuries in the Balearic Islands. I think this is everything I wanted to tell you. Thanks for your attention. I would like to thank to the team behind this. It's not my work, it's the work of many people as you, that are a big team. So behind this study was a big team, mostly from Barcelona, but also there were many colleagues from Italy as well, collaborating in this project. And I want to thank to everyone. Thanks very much.
Thank you very much. Thanks a lot. This was a lot.
A lot of information, I know. Sorry.
Information, but also I think very interesting for different reasons to various people in the room and hopefully online. So please, questions, comments.
I have many questions. One of the things, I mean, this was, like Gina said, a lot of information, great things happened here. And it's connecting to the last thing where you said it's teamwork. A lot of the things you said was like, we know, we did, you emphasize the we all the time. And from a bit more of a sociological point of view, I'm very interested in the question, who is doing what? For example, in this third tool that you were talking about, you were comparing measurements of corals with measurements of a certain chemical isotope. Is this the same kind of people who do the chemical work and who do the, say, coral work, the biological work, or is that two different groups that are meeting at certain places but are separate disciplines?
This what I presented was the work from literally. I mean, those data, they were produced by researchers. They were in this project, this RRC. And in each, if you look to the presentation that you have and you can look carefully, I always try to. There was a reference, so there was like a leading person. And the first author will be the leading person of this analysis because this person performed literally the analysis. But there's always et al. Because there is a team behind. For instance, in the case of the corals, Maria de la Fuente was the person as a postdoc hired by the project. And she literally did the measurements, the uranium thorium in China. Carbon-14 in Cambridge and in Zurich and in the States as well. But there was, in the team, there were biologists which actually provided the samples. And they did the actual sampling, and they were the material that they have previously done other studies more exploring the species of corals, the age of the growth of these. They already know more or less when these coral reefs have been growing. They have already some information, and there were several previous studies or research done to understand the behavior of these coral reefs. What was very new, it was the very first time that anyone performed this combined uranium, thorium, and carbon-14 in these corals. So that was new information we provided, and it was the very first time that this very old water masses was detected. So that was the new information what we provided, but obviously without the work of many people before, which is acknowledged because they had been in the publication. And actually, what in our system usually the leading person is doing the writing, usually because the project, the starting ideas, they were coming me. So the writing is done. I involve very much in the design of the paper and the discussion. And when it's already developed, we share with the rest of the team. And some of the people in the team is more helping with the quality of the measurements, making sure that the equipment is good calibrated, and they know a lot about the measurements, but they won't know about the interpretation. Others are more in the recovery of material because they knew about this coral reef, but they don't know so much on the measurements we are doing. So this is because it's a team effort. So it's the leading person, which usually is the first author of the manuscript, is the person who knows more about this, is the direct involved. But then there's a team, people, because there's one more expert in some of the measurements, or more expert in the conceptual idea of the experiment, or more in the equipment of the machine, and that's the team effort. A single person by their own, it wouldn't be able to do any of this work. And that was the case of the coral reefs, but you can extrapolate this for each of the subjects. Has been like a leading person taking care of the measurements, but a team around helping with the measurements, training for the cleaning, training for the. I mean, when you go to chemical analysis, there is always people around helping with them.
That case is very specific because it includes so many different things: biology, oceanography, the chemistry, the calibration of the measurements. You always do, no matter what, but the knowledge coming from the other fields.
Yes, I mean, and this team, for instance, Maria de la Fuente, the person who did the corals, she is biologist. Leopoldo Pena, which is a colleague, as a lecturer in my university, he was the person who my right hand setting up the lab is a specialist in radiogenic isotopes, and he's a oceanographer from in Vigo. Sara Camdarros, who is working with the MOL, now is doing her PhD now with the MOL signal and transferring the sediments. She has the double degree with the environmental science and geology. And the person who did the Paleocene record I show you, she comes from environmental science. I mean, they are really very different disciplines. I mean, one of the persons who the responsible for the mass spec, who has tuned all the methods, have set up for the neodymium isotopes, for the, I haven't shown here, but boron isotopes, other isotopes. He's a geochemist a chemical person coming from Alicante, and then he was in Paris in the nuclear agency during many years as a postdoc, and then we captured him to be the technical person just for the machine. So they are really different disciplines. Yes.
Thanks.
I don't know.
I've got more questions, but if anybody wants to follow up on this.
Okay. Yeah, I'm curious about the surface water masses that capture oxygen and other gases from the atmosphere, and then they sink into the depths. And they're key to understanding the initial surface conditions in which those water masses were formed. So I guess what happens in the deep oceans can be interesting to atmospheric physicists and to atmospheric scientists in general. So I wonder if these kind of connections are lately being traced and figured out are prompting more collaboration between atmospheric scientists and oceanographers. I think my question is kind of in the line of what Gibbs said before, because acknowledging that the climate system is complex doesn't necessarily mean that our scientific community is going to actually cooperate more to understand those complexities. Normally, it's the model, the driver of this communication. It's where the interdisciplinarity occurs, but that doesn't mean that those communities are close together. So yeah, what are your thoughts?
Yeah, well, it's more a reflection or the thinking about this connection. It's my own experience, which is, I think, is different from other centers where they have more modeling implemented. But for me, it has been always hard to work with physical atmosphere, because the time scales, physical atmosphere, the time scales are so fast. Changes in the atmosphere are so fast, and we are talking about time scales that they occur, changes that occur more long time scales. So I think that has always difficult in some way this connection. I have to say that telling this, I have very good friends, colleagues in physics that I think that for many years we keep saying, we need to do something together. We need to do something together. As in my university, completely constantly, there is potential to work together. There is potential, but at the end of the day, what we need is funding to have the excuse to have a project together, and it's not so easy because in the way that our agencies work here, at least, is that the projects, the national projects, it's not so easy to do projects with other institutions, even though there's departments, because there is only one partner that receives the money. And so the way that the system is set up, the Spanish system is set up in such a way which is not so easy to interact with other institutions. And in the European system, it's another way around. It's set up to collaborate, but with other countries, with people from other countries, but not from people near like you. But sometimes when you need to tone your field is so different. Your vocabulary is different. You really need to sit down and discuss a lot to see where you can meet. And I think that really to be able to really collaborate, you have to be close and be able to talk one day, to let them to understand your data, you understand their models, and is one thing that you cannot do in remote, I think. And from my point of view, I will need some sort of funding that will let to work with close physical atmospheres to trying to understand better the synergies and the potential to collaborate more.
So would you say that the lack of time and funding leads to different conceptual understandings of the data and of the ocean?
We always move in the comfort area. All of us, we do. And to be pushed to I mean, at the end of the day, our time is committed with lectures, with our own projects, and to do one thing extra, which is not part of your lectures or part of your own projects, needs an effort that you want to do, you wish to do, but you never find the time to do it. Often, ideas generate having a student or master student, someone which can implement these ideas, do tests. For that, you need a sort of background, which often is a project which pushes you to do this initiative.
Mm-hmm.
In the way that our system, the Spanish research system, is set, is just to keep in your comfort area. Is not letting to promoting that you mix with other areas.
Ayman, would you have this?
Yeah, thanks very much for this detailed and extensive way on how we measure the circulation in oceanography. You are focused on the way how to do it.
Mm-hmm.
However, I'm more interested in more about what is happening around this display. My question, could you briefly mention what is the main drivers, real drivers behind studying circulation in the Mediterranean Sea or in general,
Mm-hmm.
and what are the implications of making this knowledge of the circulation of the Mediterranean Sea? Is there any changes in the drivers in the past and in this last century and now, why they are studying now, and if we distinguish between periods or time? Also, I have a question about, you distinguish between surface circulation and deep water. What do you mean by surface? What is the surface range? What is the deep? And then my question, how do you rely on bathymetry field, or there is any relation between bathymetry and ocean circulation? I mean, this field is important to know the circulation of the Mediterranean Sea. And yeah, this is, and the last question is about sedimentation. Just to clarify, sedimentation is under which research area? Is under geology or still under oceanography or history?
Many questions. I'm not sure if I forgot. No. The first one, if I understood well, it was motivation, the driver to the motivation. I think that as I mentioned at the beginning, Mediterranean is very sensitive because we have this small box. It's easier to change, and we see that when we look the past record, changes in temperature have been far larger than in the open ocean because it's easier to cool or warm this. So during my PhD, I could see that the Mediterranean had this capacity to respond very quick to very fast climatic changes. So I got here very much motivated to understand how these changes were propagated to the deep Mediterranean. I could see that through the atmosphere in the Gulf of Lyons, there were strong changes in the convection in the Western Mediterranean Sea. I concentrate very much in the Western Mediterranean Sea. But nowadays, my question is, I don't want to see the Mediterranean as a separate boxes, Western, Eastern. So my motivation here was to see how the different parts of the Mediterranean could be connected within them, if there was any connection or not. And I look in the past time. I didn't mention here, but it's, as you suggested, probably there are moments in the past that the forcings has been different, and that's right. As I mentioned before, the Sapropel one, there was a forcing, but we have other moments in the past that I didn't mention now, like the Henrich one or the Borinale, where we know that there have been changes in the Mediterranean, and the drivers can be different, and the consequence difference. So I mean, my motivation was to understand how different drivers impact in the Mediterranean and in the different cells, whether there is a cell which is more sensitive to these drivers or to the others. And so that's a bit the motivation, is to see how different climate changes with different drivers impact in the overturning of the Mediterranean. Looking to the future, we have a situation of climate change where there is not the perfect analog in the past doesn't exist. There's no exactly the same analog, but there is some, each of these times can be some element which is in common. For instance, I have mentioned a lot this Younger Dryas period, because it was the big surprise in the project. We were not expecting to identify so many changes, but it has been in the last IPCC report. It was mentioned the Younger Dryas as an analog of conditions when, in the case that the AMOC system, the AMOC, this current system in the Atlantic Ocean, which is expected to weak by the end of the century, but there was some, could be the possibility that arrives to slow down a lot, even shut down. So the Younger Dryas is regarded as one of the periods analog to a shutdown of this AMOC. So from that point of view, it's interesting to see what was the reaction that had the Mediterranean to such climatic event, even though that wouldn't be exactly in terms of will be analog in terms of the aridity and the salinity. Potentially temperature wouldn't be so analog, but it's interesting to trying to understand what was the response of the system. So that's one of the motivations. There is no whole answer, but at least partial answers that we can get going to different time slides in the past. I'm not sure whether I have answered your first question here.
Yes, maybe you misunderstood me about the drivers. Drivers about why we are studying circulation, and the driver that you mentioned is about the change itself.
Yeah, to understand
why we need to study circulation, and what are the implications if we have this knowledge.
Yeah, I mean that would be is to understand the sensitivity of the Mediterranean to climate change. Oh, that would be my driver. I was talking about climate drivers. Yeah. And the definition, you were asking me about the definition of the surface. My definition is very much based in the water masses. For me, the surface is the circulation of the inflowing water mass coming from the Atlantic and transferring through the Mediterranean Sea. That progressively is changing properties, is getting modified. So when I talking about surface, is usually is referred as the layer, the first 150 meters, 200 meters. It depends on the basin. It's not like but this is the surface layer. Is the layer which is mostly formed by waters coming from the Atlantic. This is what at least I refer with surface. It's not purely surface, because surface will be the what is in contact with the atmosphere. But when I talk about surface in the Mediterranean, I probably prefer more this concept of this inflowing water mass. Probably every time you should redefining what you are talking about.
It's not about distance, but
No. But it's true that when there is topographic accidents are going to alter the circulation, and this layer can become thinner often. So topography is going to have an impact in the thickness, in the speed, in the position of this water mass. But I'm defining it more from the oceanographical point of view, the properties of the water mass, more than from the point of view of strict depth or deep. One of the things I'm discovering is that we talk about deep, intermediate, but potentially in the past we have got different intermediate waters, not only one intermediate waters. So deep is a bit abstract. Sometimes I'm referring really at the very deep one, the below 2,000 meters, but sometimes I'm referring to anything below Now in the talk, I've been not very strict defining this deep. Obviously in a paper you have to live very clear. Now I'm telling how the deep like this depth. It depends very much. At the moment we are discussing and the convection that we are talking, it depends a little on the moment. You mentioned about sedimentation. Of course, we're using sediments to reconstruct past oceanography, but to make sure that a sediment record is good to do paleoceanography or paleoclimatology, you need to understand the sedimentology. Means the sedimentary processes, the characteristics of the sediment, which is a discipline which is not paleoceanography, but is close to it. Usually are more geologists what define this sedimentology.
It's under geology?
It's more under geology. But I consider myself geology. I mean, I'm describing, it's also my field. I know that some of the people who work with me, which are more oceanographers or are more biologists, they don't really know so much the sediments. But any sediment core that we are working, I have been trying to be able to do the sampling myself to make sure that I can see the sediment. I can do measurements to check the continuity of the sediment, if it's good or not for sediment, for paleoceanographical. So it's more sedimentology. There are some geologists that they do sedimentology and they don't do paleoceanography. There are others who do sedimentology and paleoceanography, and there are others who do paleoceanography even though they don't do sedimentology. But I've been working a lot with sedimentologists, which they do classical sedimentology. I collaborate a lot with them.
Sure.
Before we go back to Jake, maybe I have a small question. Well, it's just maybe as a general. So it's more an oceanography question than maybe paleoceanography question, but it relates directly. So to frame it in a very general way, maybe very didactically, could you explain what is the effect of life upon oceanic geochemistry? So you mentioned photosynthesis and probably phytoplankton having to do with some of the parameters that you are studying. So just for people who maybe don't know, how does life affect geochemistry? And then you mentioned, for instance, when you mentioned the proxy 5, number 5, there you use for foraminifera shells, but the role of life is not only about as a proxy for the scientists, they are also affecting the actual composition of the waters and therefore probably the physical oceanography of circulation, right?
Yes. Life, I mean, as I mentioned, life is going to control very much the chemistry of the ocean. It's going to control it. Obviously, is the source of oxygenation. Life is responsible. Also, from the carbon cycle is going to be very important. And the carbon cycle, and nutrient content is going to be very, very important. So one thing is how life is going to be controlled, the chemistry of the ocean, and it determines life in the upper ocean. Is photosynthesis which is dominating these changes, but which is the water mass enters in the ocean. It's going to be more processes of degradation of the organic matter according with the flux of organic matter. This intensity of degradation is going to change the chemistry of the water mass. So life and biochemical processes have a strong effect in the chemistry of the water mass. It's going to control the pH of the waters. But then of course we use life to do our reconstructions.
Sure.
We use, because as you mentioned, we use a lot these foraminifera shells for our reconstructions because they become stored in the sediment. We can identify, they are great, and so it's absolutely true that we need to understand the biology of our carriers, because these are the carriers of the signal. But to understand the signal, you need to understand the vital cycle of these carriers. So we need to know whether the specimens we are picking in the define, where do they live? And some of the species, they live in the very shallow waters, very surface. Other is in the deep thermocline, but others, they live, they are benthics. So before interpreting our data, we have to be very sure which species are we working with is the environment, then seasonality, because some of the species, they live in spring, others live in summer. So according with that, we can really say, well, we have here the spring signal. And that's the reason why sometimes we can compare different proxies. They are not all in the same, but it's because it's true. They are based in carriers that they have different vital life, and so they have a different environment. So part of our job is really to understand our carriers and the life cycle to really understand the signal.
And then two small questions, maybe. The first one, you mentioned mostly 25,000 years, that time frame. Do you go at all the way to the Messinian crisis, or you don't need to?
Well, you don't need it.
Well,
I mean, I think it's very interesting, the Messinian crisis, and it's a very also challenging period, a very interesting, but at the end of the day, you have to focalize in targets, because working at this resolution I'm showing to you, to go down to the Messinian will be several lives to be able to do it. So I know I haven't been working with the Messinian period. I know other colleagues, also because probably you don't know, but we didn't have marine records down to the Messinian because we were not allowed to drill them because it's highly dangerous. So the oceanographical vessels which are able to drill down to the salts, they were not allowed to go because the salts are very expensive, and so they can kill everyone on board. So the ODP program was not providing the permissions to do that, but it's going to happen this year, I think, or probably it has already been done recently, with a Chinese vessel is going to be able to drill down to the Messinian. So we'll be able to work with that. Other colleagues more specialized on those periods. In my particular case, my work has been mostly done for the last deglaciation, also during the glacial period, the rapid variability of the last glacial period, which is the last 50,000 years. But I've been also working for the last 3 climatic cycles, which are over 300,000 years, that more for the Pacific Ocean. And in the Pacific Ocean, I've been working for the last 2 million years as well. Of course, to have that, you need material from vessels that Spanish vessels, they are not able to drill such a long sedimentary sequences. But with some of the platforms we have from international projects, it's possible. But it's a matter of time. I mean, there is many interesting questions to address with these long sequences. But my career has focused mostly in this recent period and trying to understand better the Mediterranean, which really fascinates me. I mean, I have to say, I've been doing a lot of work in the Pacific, also in the Atlantic, but I feel like I'm a Mediterranean person. In the terms that I know it well, and it fascinates me, and there are so many interesting questions to address. So I love to work in the Mediterranean. So I went out of the Mediterranean and then I went back, and I'm happy to be back again in the Mediterranean.
Did you have more?
Yeah, I got one follows up on what you were asking before. We talked about that yesterday, actually. From what you said about the need to know about the biological life cycle of the carrier that your signal that gives you your signal. It's very interesting that you seem to be talking about cases where you know the degradation of the signal very well, and it seems to be that you talk about cases where the life is actually dying off. Is that a thing you can generalize? Is it to what extent? I mean, let me rephrase it in a more interesting way. To what extent do you actually need to know about the generative processes of the signal? You rather not have them, I suppose. You rather have a signal that is really clear and that does not regenerate anything like that.
I haven't known in my presentation because there was already too much information, but I didn't put any attention on the preservation of the signal. Mm-hmm. But this is a big issue for us. It's a big issue because I've been telling you that the magnesium calcium is a ratio for temperatures, but there are many factors that can alter that signal. Not only the biology of the carrier, which is one you need to know it, but there is other factors, diagenetic factors, the overgrowth of diagenetic carbonate, the appearance of clays that can pollute the signal, the processes of dissolution that they produce partial selection of the magnesium-rich carbonate. There is many factors, and you will get very bored if I told you about them. So it's the reason I didn't put it in my talk. But it's true. I mean, we need to answer to many of these questions. So there are different ways. For instance, I always been very obsessed about this subject, I have to say. For instance, the very first PhD that I supervised, the main budget and the main target was to find these problems, the unique problems, using laser ablation over the foramifer to explore how the chemical signal was across the shell, to see the genetic processes, and to explore how the cleaning process could remove that. I almost failed by my previous supervisor at Cambridge, Harry Delfield, a very good chemist, but I changed the cleaning procedure that I learned in Cambridge to make sure that I was able to get rid of pollutants in the signal. So you have to be a lot of care what you do, and needs to be checks in the cleaning procedure. I try to do same pictures to see how the surface after cleaning is, whether it has been removed, the clays and secondary calcite. But at the end of the day, in palestrinography, I would say the best and the thing which makes you more confident that you are on a good track is when you can reproduce a signal. If you can really do another core nearby, and you reproduce the signal, and that happens very often. Sometimes no, and say you forgot about that because there are factors. But when you see that you reproduce a signal, for me is the strongest proof that you have something interpretable. But for us, it's a very, let's say, artisanal. It's very, I mean, to have a record is a lot of effort, and it's very expensive, and it's a lot of time consuming. So it's not like we can reproduce now. I mean, it has done. I did the record I did for my PhD that was highly discussed whether I was able to see changes in the North Atlantic into the Mediterranean was highly discussed, and I fight very hard to tell is the truth. Now has been reproduced by many other people after that, and it's the same signal. It's perfect. It's amazing, but it took years to be reproduced that signal. So the best we have is when we can reproduce it. But of course, there is ways to trying to make the best you can do of your job. That there is always other factors that can occur.
Connected to that, most of the tools that you use, that you explained to us, use two signals. One that is a chemical thing that relates to whatever you want to study, oxygen or whatever. But then you need something to calibrate.
Yeah, we cannot always calibrate. That's the ideal one. You have a proxy. With the proxy, is no proxy means it's an indicator of environmental parameter. Ideally, we would like to have a proxy which is quantitative, which means has been able to calibrate. And we do have that with temperatures. We have these calibrations, obviously with errors. But I mean, it's pretty good when I can tell you that we know the ocean temperature 100,000 years with plus 0.5 degrees. I mean, it's pretty good, but we have always an error. But temperatures, we can do quite well. But when we go to other variables, I told you about salinity, but I didn't put absolute numbers. That's a qualitative. It's saltier, fresher. We could try to do it. I will try to put salinity values, but I will never trust myself doing that because the amount of errors we do have in that translation is so big that I believe on the relative value, but not on the absolute value. And with the oxygen content, there is no calibration, but I show you a sort of calibration, which I think that since it's a proxy relatively new, there are not so many studies. I think we are still far from an absolute calibration, but I think orders of magnitude is good to say that there is. I wouldn't say precise orders of, but with an error bar, more or less orders of magnitude, but it's not precise calibration. But often we have to live with more qualitative proxies than quantitative.
And to what extent does this play a role in the question of funding that you were discussing earlier? Because, of course, you would rather have the best information, but I guess it takes more time, takes a lot more work, you need funding.
No, but I'm not complaining about the funding. I mean, we have good funding and I have got very good projects. What I was complaining about the possibility to do interdisciplinary funding with other disciplines. In the way trying to more link our disciplines. But from the point of view, I mean, from the European side and from the national side, I have got very big projects. So it's not that I'm not complaining they're not be funded, but we're saying trying to do more interdisciplinary developments of the science.
Yeah.
Sure. The time is scary.
Sorry?
The time is scary.
It's a limitation. I absolutely agree. The time is, but I think that nowadays the models have really progressed very much in the sense that they are now they can assimilate centuries, and we have the capacity really to do several thousands of years runs, and that we have improved the resolution, our records, and the time resolution. So we are now in a position that could be closer to trying to define common targets or strategies to really interact more. But I mean, the first limitation is this time scales. I totally agree. But I think that even though there is, I mean, in some institutions in France that the models are closer to, I think they are interacting more than in Spain and France, for instance, and in Germany, I think.
I have one question for Nicolas. Is that acceptable? Nicolas, can I ask you a question?
There was a question here.
Let me ask very briefly, because I already called his attention. Do you also use, as an underwater archaeologist, carbon-14, or because of these differences between how it behaves underwater, you don't use it?
I personally didn't use this kind of technique for dating, but is usually used. Yeah.
Of course, in surface, everybody knows that archaeologists use it, but also in marine archaeology.
It remains, but there is this issue you mentioned.
You need to correct that for reservoir age that is apply a lot.
But the error is systematic, so you know how to correct. Sorry, Jose Dario. Alberto, do you think we will hear him? Or does he need to come closer? Acércate un poquito al micro, por favor, que estás ahí lejos. Perdóname.
Well, there, right? You're telling us about, well, if I got it right, about a very different period in history. We're now approaching possibly a change, more or less dramatic, but in any case affecting many systems also in Earth. And you're telling us about the Green Sahara, last Sapropel event. I don't know in which order, Sapropel, Younger Dryas, and all that. But I didn't, because your talk didn't focus on that, see the place of life in all this. This is a different question from Linus, but I would like to see how much of a paradise or a hell does this period you are examining for life, because the Green Sahara sounds very good, but for the Mediterranean marine life, how was it?
This Green Sahara period was the period of the Sapropel one, and after I did, but I was discussing about changes in the convection. I did that there was, but also there was an increase of productivity, marine productivity in the Eastern Mediterranean Sea. And that's believed that this increase in productivity came because these rivers provided more nutrients to the Mediterranean. So it was a primary productivity increase that is very well identified as well with barium concentrations, with barite, which is a mineral which is synthesized by organisms. And so it was a period also of high productivity in the Eastern Mediterranean Sea. In the Western Mediterranean Sea, we don't really identify any substantial change in terms of productivity related to that period. But in the Eastern, yes, there was more marine productivity.
It's interesting because I recently read something about there was a misconception before. There used to be a misconception about the Eastern Mediterranean being a boring sea, being a dead sea in ancient science, I guess. But now becoming more widespread, the idea of being actually the interesting part of the Mediterranean Sea, right? So your research indicates that.
Yeah, I found really, as I show you this picture of sediments with so many changes of color. Which is amazing, really. There was so many changes. There was a Sapropel, but I think it was a very dynamic system. And I think we don't fully understand all the changes that occur in the Eastern Mediterranean. Still, I think in some way they were also extended to the Western later on. But I think that is really the source of many changes. Changes in primary productivity, moments that there was really high productivity. And of course, those Sapropel for the deep layer, the life disappeared because it was dysoxic. So benthic life disappeared, but planktonic life was very alive. Now always the flourish of life is on the same level for everyone.
Please.
So I had also a question about calibration, so you partly already addressed it. But could you maybe say more explicitly? So for the cases where you can calibrate quantitatively as with temperature, if I understood correctly, so what is what you use to calibrate? Because you do not use what you have learned about temperature in the past. I guess maybe you use what you have learned from other proxies. And then a follow-up question is, for the cases where you cannot calibrate quantitatively, how much uncertainty would you say you have in those cases to then interpret your results? So it seems that for each proxy, no, you refer to kind of interpretation stage where you actually come up with an outcome of how to read this, no? And I guess in these cases where you cannot calibrate, how much uncertainty is there in the air, or maybe it's not so serious as sounds.
Okay, this is a very good question.
Very general.
Yeah. For temperature calibration, there are two main strategies that is used. The first one is with culture calibrations. Culture means that it's possible to grow some of these organisms in tanks. In the case of foraminifera, their plankton, you can fish them and then grow them in tanks with control conditions of temperature, but also the parameters. You can change salinity, you can have different. So, and then when it ends the vital cycle, you measure and you have exactly the conditions in which they were living. Three ways. One is with the cultures. The other one is with sediment traps. Sediment traps are these traps of particles we are following. They are settling down in the water column. So sediment traps you can really program to close the bottles every 15 days. So you capture what comes down to the water column during 15 days. So you capture foraminifera, for instance, there, and you know that that was February within 15 days. So you know what was the temperature conditions during this time. So you can calibrate the chemical composition of those cells that were settled down with the temperatures, satellite temperatures, or hydrographic temperatures you might have. And the third way is taking core top samples, which means surface samples in the surface samples measure the chemical signal and calibrate with the temperature at that location. That's assuming that this surface sample is subactual or actual, and you calibrate to the surface temperatures, because sometimes there are sedimentological processes that have cleaned out the recent sediments. But if you can date those sediments that they are recent, so you can calibrate with the core tops. So these are core top calibrations, sediment trap calibrations, and culture calibrations. There are three strategies. If the wall is perfect, they agree, and they give you the same calibration. Which actually with the alkanones, which is the proxy I use for my PhD, with this organic biomarker, is fantastic. We have a unique calibration for all the oceans, and they agree pretty well with the different ones. It's a linear calibration. With the magnesium calcium, the life is a bit more difficult because it changes a lot from one species to another species. But we have an exponential calibration. It works pretty well, but we have a larger uncertainty than with the other proxies. That's the first question. The other question is the other proxies. I mean, it will depend very much of the proxies. Most of the times, people is just showing this non-calibrate, more objective, more relative proxy, you say more faster or speeds or slower speeds or more oxygen, less oxygen, so it's more qualitative proxy. But I always, when it's possible, I like to go to trying to learn a bit more. So that's the reason for that experiments we did with the sedimentals for the speeds, that at least gave us a framework that what with some speeds we could see the maximum size that it was possible to the maximal size of the sediments we collected. So at least we have a boundary values that we know that one speed is able to transport this. So we won't be able to quantify precisely the speeds, but we have like a framework to put some limits. But it will depend on each proxy and the how much has been an effort behind trying to compare with current conditions or not. Thank you.
Just a question. I have read that the total volume of the Mediterranean sea water can be exchanged with the Atlantic Ocean in 100 years, 100 and some years.
Mm-hmm.
So in all the studies of oceanography and circulation, they look into consideration this thing. I mean, you are analyzing the water, maybe it's not old waters.
Yeah.
I mean, the result of this study and this water now is coming from the Atlantic Ocean 20 years ago. I mean, how do you...
Well, one part of the proxies was mostly and the one to see reservoir ages was to see whether this has changed in the past time or not. We consider that we have the inflow of what when it's in surface, when water is at surface, we always consider it is a young water mass because there is always exchange with the atmosphere. So that's a young. It's a no age. Is a recent water mass. It will exchange properties with the atmosphere, and the properties will be changed in the path. But what in the moment it's going inside the ocean when it becomes isolated with the atmosphere, is when this water mass can become older and older. Is the time that water mass is insulated from the atmosphere. And nowadays, as you mentioned, this isolation is the order of 200 years, something like that, 200 years.
But this is a fact, not hypothesis.
It's what more is the deeper ones is what has been estimated is this order of magnitude. But it has not been always like that. That was my hypothesis, and is what we have proved with this technique that I showed that dating these corals with uranium, thorium, and carbon-14, that in the past time has been periods of time that they were over 1,500 years. So
they don't change.
Well, the age of that water mass was 1,500. It means that has been for a long time inside the Mediterranean without going out in somewhere. We cannot tell you with our information whether that was stored in the western or in the eastern, but we know that for a long time was an storage of water mass. So probably was I believe it was more intermediate depths store, and there was probably deeper and shallower waters going out. But in general, there was a slowdown that occurred during the deglaciation, a slowdown of this renovation. And so for longer times in the Mediterranean. I'm not sure if I have answered your question.
Yes.
But
I'm not an expert in the field, but
so at the moment that you have a water mass for longer time, so the chemistry is going to change. It's going to change. The chemistry is going to become depleted in oxygen because the organic matter which is falling down is going to consume the oxygen, will become rich in nutrients, will change the carbon signal, will enhance DIC, dissolved in organic carbon, but at the same time the alkalinity will become lower and become more acid. So you change the carbon system of this water mass when you have it for so long. So for that reason, we believe that water mass which is passing through these coral reefs is so acid. This water mass has such a low pH that doesn't let these coral moss to develop. Coral moss.
Okay, I think we should leave it here because we've already been using you for many meetings before, and this was a long Q&A anyway. We can continue the conversation over coffee or beer or whatever everybody wants. So thanks a lot to everyone. Thanks to the people online and later on watching on YouTube. Thank you very much, Isabel. Muchas gracias.
Thanks very much to you, Paco.